What does surface roughness mean for medical implants? Clinical inserts are gadgets made to supplant, help, or work on a weak organic construction. Frequently, they are created as prosthetics to supplant missing appendages and body parts. Another normal utilization of inserts is for the conveyance of medicine, the checking of different real capacities, and offering help to tissues and organs. Around 7 million Americans have counterfeit joints, implying that essentially 2% of individuals in the US have a clinical embed. Clinical inserts are set inside the body or onto its surface. Consequently, the manner in which they cooperate with the living tissue is fundamental to the manner in which they work. The synthesis and surface harshness of a clinical embed incredibly impacts the pace of osseointegration, bone ingrowth into a metal embed. Studies have shown that unpleasant surfaces give positive conditions to osseointegration, supporting organic exercises with biomechanical dependability. Here, we examine exhaustively what the surface unpleasantness of clinical inserts means for their usefulness. Studies show that surface roughness of medical implants improves osseointegration. Throughout the long term, various examinations have exhibited how embeds with surface harshness in contrast with those with smooth surfaces advance more noteworthy degrees of osseointegration. In one such investigation, analysts analyzed the bone redesigning action on two various types of titanium inserts, one with a machined smooth surface and another with a plasma-showered harsh surface. The group utilized a confocal laser filtering magnifying instrument, CLSM, to gauge the degrees of direct bone embed contact and bone volume after the inserts were installed into the femurs of bunnies. Clinical-grade adhesives and sterilization processes. Surface measurement is crucial for high-quality and reproducible medical devices. How white light interferometry can measure surface roughness. The outcome showed that the harsh surface titanium inserts advanced more noteworthy rates of direct bone embed contact and bone volume in examination with the smooth surface titanium inserts. They additionally discovered proof to recommend that this expanded bone volume saw in bunnies who had the harsh surface titanium inserts was expected to less redesigning action during the beginning phases following implantation in correlation with the smooth surfaced inserts. The group inferred that the surface harshness of titanium assumes a critical part in deciding the fragile harmony between the development of new bone and the resorption of rebuilding at the interface of the inserts. In later years, researchers have endeavored to additionally work on the achievement of clinical inserts. Examination into clinical inserts has started to give extraordinary consideration to the viability of inserts with miniature harsh surfaces. Inserts that use miniature harsh titanium surfaces have acquired consideration, and their utilization in clinical inserts has expanded in the course of recent many years. Discover more. Physical properties testing equipment. Inserts with miniature harsh titanium surfaces are generally made by means of diminishing methods, incorporating coarseness impacting with aluminium oxide or TO2 particles, a blend of sandblasting and corrosive carving, or corrosive drawing alone. Different in vivo contemplates utilizing distinctive creature models have tried the effect of these surfaces on the embed's viability at advancing osteointegration. In evaluating different examinations that research this more current clinical embed material, it tends to be inferred that inserts with miniature unpleasant surfaces actuate GON mix in more limited time periods and accomplish a higher level of bone embed contact, just as a more significant level of protection from shear in correlation with inserts that have a smooth surface. Researchers have investigated the hidden systems that permit surface harshness to adjust these impacts. Later in vitro exploratory examinations have revealed some insight into this by researching reactions at the cell level to embed surface geology. The information gathered from these examinations has uncovered that osteoblasts, cells liable for the development of new bone tissue, are touchy to surface unpleasantness. Osteoblasts presented to surface unpleasantness show diminished multiplication close by a more separated aggregate. What's more, PGE2 creation has been demonstrated to be increased on harsh surfaces, as is TGF-beta1 creation. This information shows that surface harshness might have the option to intervene the autocrine and paracrine guideline of osteogenesis. At last, contemplates have shown that surface unpleasantness can viably tweak the impact of foundational chemicals, for example, 1,25,O,2D3, on osteoblasts. Clinical preliminaries have additionally shown the advantages of delivering clinical inserts with unpleasant surfaces instead of smooth ones. Clinical preliminaries in people have shown that roughened titanium inserts require a diminished mending time period prior to stacking, 6 months rather than the typical 12 weeks. More limited recuperating times convey numerous clinical benefits. For example, more limited medical clinic stays and diminished misery, sway on life for the patient. Information has shown that the surface unpleasantness of inserts can imply that more limited sizes can be utilized, 6 to 8 mm, than those routinely acknowledged. The utilization of more limited inserts offers the advantage of keeping away from the requirement for more broad surgeries, for example, sinus joining in the maxilla and nerve lateralization in the mandible. While more examination and information are expected to permit us to completely understand the most extreme capability of utilizing unpleasant surfaces on clinical inserts, the exploration presently focuses to the various advantages of surface harshness, especially for the acceptance of osseointegration.